So before I start, I'm hearing impaired, so if I'm swallowing some of the word or not answering correctly, please, I do apologize in advance. So today we will speak about hydrogen, and I will give you some background. And I will give you background from the view of the customer, because I think it's more understandable that way. And then I will briefly talk about the science that we are trying to do, because people here are coming from such a diverse field, so I will try to make it interesting. So the goal, as Itai just said, our goal is to do molecular farming. We have an amazing enzyme that you can find in many different kinds of organisms, in microalgae, in bacteria, and that enzyme has this catalytic site, which look completely inorganic. For us, biology, we see a lot of toxic material here. We see carbon monoxide here, we see cyanide here. But the enzyme that holds this thing in the heart of it can produce hydrogen at an amazing rate. And the rate is 9,000 hydrogen molecules per second per enzyme. The only substance that can produce hydrogen at such amazing rate is platinum. And all of us know what is the price of platinum. Here we are speaking about an enzyme that anyone can produce in E. coli system, et cetera, et cetera. So there are two routes where we are trying to exploit this enzyme. One route is with Lee, and Sachi, where we are trying to do in vitro system, nanosystem, that will feed this enzyme with electrons. The other route that dumb biologists and myself do is try to convince microalgae to use sun power in order to provide electrons to those enzymes. But before we go, I will try to convince you why hydrogen in the first place. So if we do it using sun power, then we have a true renewable energy source that is produced by sun energy, and we can produce it almost anywhere on the planet. So it can be the most socialistic way of energy production. The second thing, that when you burn hydrogen, either directly or by using it inside fuel cells, you emit only water, just water. You have no carbon footprint. The other reason is that hydrogen has a huge dense energy. If you compare a kilogram of hydrogen and a kilogram of gasoline, the kilogram of hydrogen has something like 50% more energy than the gasoline. And it can be produced anywhere on the planet, I just said that, and it has growing market. What kind of market? We are not speaking about Haber process and other things that any chemist knows. We are speaking about transportation. So in 2015, Toyota started not a fancy project, started a mass production of hydrogen-driven car. The name of this car is Mirai, future in Japanese. 5 kilograms of hydrogen inside this car give you a range of 500 kilometers. Fueling stations are being built in Japan, in Australia, in America, in North Europe. So society starts to get the idea that a new kind of transportation based on hydrogen is something that we should support and provide. Personally, I don't believe in cars at all. I believe in electrical bicycles, and anyone who go in Tel Aviv street can see the revolution in the front of his eyes. The best way to see that is to go to high schools. In each high school, you see hundreds of electrical vehicles. I don't believe that those people, when they will have kids, and myself, I have 4, by the way, unplanned. Anyway, I don't believe that those people will use automobiles because they are growing with the bicycle, with the scooters that you can land anywhere on the street. So 33 grams of hydrogen in electrical bicycle give you a range of 100 kilometers. The most awful fear that I have with my lithium battery bicycle at the moment is when it will stop. And the range that I have is something like between 20 to 25 kilometers. So while in automobile industry, we simply replace the same range, 500 kilometers with gasoline, with 500 kilometers with hydrogen, with bicycle and scooters, we really have a breakthrough of technology. Now, let's speak about producing hydrogen using sun power. Sun power that arrives to 1 square meter in Israel is it's something that is very constant. And if you convert it by, for example, by current technology of photovoltaics and electrolysis, 16% and 0.5% efficiency of electrolysis, you can get something like 4 kilograms of hydrogen per square meter per year. That technology exists today, but it's too expensive for upscaling. The second technology, which is not exist, and it's just a dream, is biological hydrogen production by photosynthesis. There, using natural photosynthesis, we can dream about almost 7 kilograms of hydrogen. So we are trying to make this from a dream into reality. The question is, from your eyes, does it worth all the effort? 1 kilogram of hydrogen gives you 100 kilometers. That means that 1 kilogram of hydrogen equals to 10 liters gasoline. You as customers, at least in Israel and Europe, are currently paying something like $17 per 10 liters of gasoline. In the US, it's about 7, but US is not an example for anything. So if you produce it something like at 66% of what you earn, you can still invest $11 in production of kilogram of hydrogen and get some profit. Not high-value product, not industry. It should be comparable to wheat, to rice, to agriculture. It's something very, very basic that we should invest maybe a lot of money and get a portion of earn, but on this time we will save the pollution that we are producing, we will save the earth, and it will be the same as we speak about food security, we can speak about energy security. So at 4 kilograms per square meter, the expected gross income per hectare can be almost $700,000. My dad, from hectare of avocado, earned this amount, gross income. So it can be a very nice way of agriculture. Many agri people will like to do this thing instead of the avocado they are producing today. And lastly, I believe that hydrogen produced by that way should not be taxed because it's a clean energy carrier and it's a basic agricultural product as apples.
Can I interrupt now?
Sorry?
Can I interrupt now?
Yeah.
This $680,000, that account also for the infrastructure, for compressing the hydrogen?
That gross income. That the gross income. I don't know how much it will cost to produce it. That depends on us, what technology we are going to develop.
No, but with hydrogen, there's an enormous amount of infrastructure.
You're absolutely correct.
It's not like avocado.
You're absolutely correct. I'm just saying that society will not pay you more than that for the 4 kilograms. So if you, as an engineer, can produce something that the production cost will be lower than that, that's fine. I'm just describing the arena. I don't know if it's possible or not possible. I'm just describing. From the algae side, I know what is the situation. What is the let's assume that we are succeed in using nanomaterials or biological materials. We are able to do these 4 or 5 kilograms of hydrogen per square meters. In the state of Israel, we have 3 million cars, and that's the amount that typical car drive. The area that is needed is 135 square kilometers, 0.6% of the area of the state of Israel, if we are successful. If we do that with a bicycle, 3 million bicycles, we need 1.75 square kilometers, a city quarter. Again, the technology, we need to, actually it exists currently, but it's too expensive, so we need to invent new technology, either nanotech, biotech, But that's the situation. That's what we face. In biology, by bioengineering of the system, I told you that we can, natural photosynthesis, we can dream about almost 7 kilograms of hydrogen. But by chemical fusion of hydrogen to the power source, to photosystem 1, the group of John Goldberg was able to get something like 11.4 kilograms of hydrogen per square meter. Why I'm showing this number? Because that's the maximal that biotechnology effort and bioengineering effort can take us. So I'm just showing you the borders of what we can get. With nanomaterials, maybe we even can get more, because nanomaterials can use wavelength that photosynthesis doesn't. UV, far red, etc., etc. So there is much to do in this area. Now let's speak about biology a little bit. When you take this green scum that you have there in open pans, and you seal that in a bioreactor in darkness for something like an hour or 2 hours, or simply darkness, that's what you see here in gray shade. When you illuminate it in green line, you see fast hydrogen production, fast accumulation of hydrogen, okay? While oxygen is slowly start to accumulate. And the hydrogen production stopped exactly after 2 minutes. For 70 years, people say that the cessation, this fast cessation of hydrogen production in biological situation is because that oxygen that is being produced by photosynthesis kills the enzyme. In bars here, you see the amount of active enzyme that we analyze simultaneously. All of this work is done in mass spectrometer and fluorescence simultaneously. And what you can see that the enzyme, when the hydrogen production ceases, the pool of enzyme is completely intact, meaning that the dogma claiming that oxygen killed it was wrong, meaning that the hydrogen has stopped to work because something simply steals the electron from it. And we wanted to know what is that something. So since we saw that we have a margin of 2 minutes in which the enzyme pool is completely completely active, we decided to do cycles of light and dark, 2 minutes, 2 minutes, 2 minutes, 2 minutes. And you can see that in each time, hydrogen production starts and then it stops at the end of the 2 minute region. Meaning that again and again and again, something is stealing the electron from the poor enzyme. And we wanted to know what is that thing. So since we are doing mass spectrometry and we measure everything, carbon dioxide, and as you know or don't know, the main purpose of photosynthesis is simply take carbon dioxide and produce sugars. That's what evolution forced the plant to do. If you do sugars, you can build yourself, you can produce oil for humans that will take it in 1 million years from now, etc., etc. And what we saw when we, that's the accumulation, but when we have carried out the first derivative of the accumulation, we simply saw the rate. You saw that once the hydrogen production rate start decreasing, the carbon dioxide rate also start decreasing. Why? Because it means that the organism start to consume carbon dioxide inside, it's taking it from the liquid, meaning we see here exactly the switch when the electron goes from hydrogen production to carbon dioxide fixation. So we found our evil entity. The evil entity is carbon fixation that stops hydrogen production. In order to prove that, we simply added an inhibitor of carbon dioxide fixation. And as you see, in the absence of the inhibitor, hydrogen production stops. But in the presence of the inhibitor, hydrogen production kept linearly. So we understand now that we need to bypass, we need to bioengineer the way so that the hydrogen will be able to catch the electron from the carbon fixation system. The question is, what is the mechanism? Why the carbon fixation can steal the electron so efficiently from the system? So if we look on the photosynthetic machinery, we have here this beautiful power plant, photosystem 1, over 500,000 Daltons, super complex of material that can get sunlight and produce electron at quantum efficiency close to 1. The most efficient quantum nanomachine that there. And all the electrons from this power plant are delivered by a single power plug of the system, soluble power plug, an entity that sits, docks on the power plant, get electron, and then donate it to the process that can get it. And here we describe an arena where a competition between the hydrogen-producing entity, the hydrogenate, and FNR. FNR is reducing NADP plus to NADPH, and that's the substance that feeds carbon fixation in photosynthesis. So either electron go to right or left. The question is why they are going here. So this entity is soluble, floating there. The ferredoxin that gets the electron also, it gets the electron and then dissociates, it's also soluble. But FNR is likely to be bound to the system. It's like a cat that's guarding the milk, guarding the electron. And using ITC, we were able to see that FNR indeed, if we have isolated PS1 and isolated FNR, we can see the binding constant, a very simple binding curve that's showing that FNR indeed can dock on the power station. It can dock on the electron source. It's simply guarding the electron and doesn't let to other entities floating out there to get electron. And that's how, when you do a kinetic experiment, if you measure the rate of hydrogen production and the rate of NADPH production, we do that using membrane inlet mass spectrometers and fluorescent measure, in simultaneous fluorescent measurement of the NADPH concentration. It was done by Pini. You can see in blue line the hydrogen production rate as a function of addition of the competitor, FNR. And you see that as you add FNR to the system, the hydrogen production rate to a minimum, while the NADPH production rate is increasing very fast. Meaning, like as we translate it from Hebrew, the FNR even not counting the hydrogenate, because it so efficiently can sit on the PS1 and catch the electron, and it's not a fair competition at all. So that's what's happening. So how we can bypass this mechanism? In another paper that we published this year, we saw that ferredoxin, this entity that gets the electron from PS1, has a multiple binding curve to PS1. The fact, actually we see here two different thermodynamic events. So if that's the PS1, ferredoxin first is bound, or it's electrostatically steered to the PS1 at very strong KD of 29 nanomolar. And then it rolls into the center of electron donation that gives us a different KD of 247. Meaning that this approaching system, we can use it for bioengineering. And how we can use it? We can simply fuse the ferredoxin and the hydrogenate and use the ferredoxin ability to approach the electron source the so efficient way we can use it to anchor or to make the hydrogenate very close to the electron source, closer than the competitor, than FNR. And that's what actually we have did in the last few years. And in vitro, we were able to see that indeed, while in the natural system, about 85% of the electrons are lost to the competing system, in our engineered system, about 60% of the electrons indeed go toward hydrogen production. Next, we wanted to do that in vivo, inside algae. And for that, we are using a special system, special reporting system, where we have a bacteria that smell hydrogen and emit GFP. So we developed a special tool that you can download, by the way, and we just published just last month. And that tool allows us to give numeric value for each of the clones that we are testing. Which of the tests is the best hydrogen producer? The larger the halation, the more the hydrogen that the clone do, but the halation are divided by the size of the colony, and so we have quantitative number where we can select clones to be tested as. So indeed, we were able to show that engineered clone has 4-fold hydrogen production compared to the wild type. You can see here. But the problem that we faced that the expression of our new nano machine, it was too low. Therefore, we had to build new algorithm that will allow us to increase. We have a factory that can hold 1,000 machines, and we have only 2. We wanted to fill the entire factory with machines. For that, we developed an algorithm where we use this fusion protein as a reporter. And that algorithm was recently published in Plant Journal, and you can use it for free from our site. You can change the DNA sequence in such a way that you will have high expression of your target, any protein, in our microalgae, Chlamydia monocyte narrative. And indeed, this work was led by Ido Weiner. The work was very successful, and in another publication that went out, we were able to show that indeed the amount of our fusion protein is much larger than the amount of the wild type here, meaning that the algorithm was successful. Now we have another challenge, because we have such huge amount of enzyme, you need some entity that will build this crazy inorganic material inside. You need 3 genes to build that. So we have a problem that we have so much of the upper protein, of the fusion protein, but we do not have enough entity to mature it. And that's what we see here. That's the total amount of protein that we have, and that's the mature protein. Meaning that now we need to build a synthetic operon that will allow us to simultaneously express all this maturation factor in order to have full maturity. For that aim, we developed another tool, and actually using this tool and artificial intelligence and empirical data, we were able to build a database. It was published only last week in nucleic acid research. And in our site now, we have a mapping of the entire plant kingdom. Any plant genome ever sequenced, we build maps of the operon there. Meaning, if you want to build a synthetic pathway, and you want to know what spacer you should put between your genes, it doesn't matter what kind of genes, you can use our database to understand from, first of all, which of the operon is the most highly expressed, so you can use those spacers between the genes that you are interested to express in order to overcome the problem. So now we are building synthetic operon, and we are trying to get full maturity, and then finally see if our route can lead us to what we want or not. And that cost me 7 years so far. Anyway, so the conclusion is that we have very tight competition in the photosynthetic machinery, and only by bioengineering bypasses, we can bypass those competition and maybe reach efficient hydrogen production. At the same time, you guys, using your nanomaterial, you can use the enzyme and hook it to other electron donors. It can be completely synthetic and get a great rate of hydrogen production, and maybe scalable. So that is my group and all the funding agency, and thank you for your listening.
Thank you, Iftach, also for excellent keeping of time, so we have time for many questions now.
They are stunned.
Do you have additional questions for me? Okay.
Can you give? So, in order that I will be convinced that your question is relevant, first of all, I want to see in small scale an algae clone or another way that can keep for hours the same rate of hydrogen production. 1 ml, if I can see 1 ml that can produce hydrogen in our term is at 200 micromole per milligram chlorophyll per hour, only then I will be convinced that this kind of question, your question is very optimistic. I'm not there. So I want to be convinced, first of all, that we actually can do that. So, you get me? So on the way, we found out that this hydrogen is an amazing tool to study electron transfer in photosynthesis. So for example, one of the things that we found that regarding the proton that you just asked, that there is some pumping mechanism that pump protons there. And we found a mutant where this pumping is occurs much slowly. And we found it because simply the hydrogen production was 3-fold higher there, meaning that the electron goes straight to hydrogen in order to, and are not cycled during proton pumping. When I saw that, I understood, okay, that probably is related to fast growing. And amazingly, under low light, this mutant is grow like super, super fast compared to other things. So now we are interested in that. So you never know where it takes you. But again, I need to see a steady rate before I will go into this calculation.
Zachi.
How can you balance when you bypass the CO2 fixation process?
That's very easy. As long as you grow them an open pond and oxygen is there, hydrogen production doesn't take place because the enzyme is very sensitive to oxygen. So you can grow them, and then you can move them to another bioreactor and then harvest the hydrogen for a week. It should be sealed one, and it should be, you have two phases. You have a phase of growth, and then you have a week or 2 weeks of production. When it's sealed, you simply starve them to carbon dioxide, very easy, minimal medium, and they cannot. Sorry?
They're still viable for a long period of time?
They can live for at least a week or something like that. So for the calculation that I've done, it's based that they will survive for a week, something like that. And then you can use it to feed pigs or make sushi or God knows what. Okay.
Okay, thank you, Iftach.